Hello, good afternoon, everyone. Thank you for coming and making the switch to online. So we are going to do half of lecture 9 today. And you know, just like a regular class, if you have any question, just um, you know, unmute yourself, or if you want to type the question in the chat box, that's okay for me as well. This session will be recorded, so if you want to watch it again later, you will have the option of doing so. All right, let's get started. Biotechnology. That is the second last lecture of the course, and as I said, it can be a little bit tricky. So what is biotechnology? Well, the name has two parts to it. There is the bio part, which is the biology, and then there's the technology. So essentially, it is technology that is based on biology. So by understanding various things that are going on in a cell, for example, scientists try to control, alter, and harness some of these cellular and biomolecular processes to create new technologies and new products that would help us improve our lives. So what kind of cellular process have we learned in the course so far? I think back to all the lectures we've done and all the things that are going on inside a cell. What kind of processes have we learned? Again, you can type the answer in the chat box, or you can unmute and tell me directly. That works probably better. Anybody? Any kind of cellular processes that we learned? Yeah, so there's cellular respiration that we learned. So by understanding how the Krebs cycle works, for example, maybe we can design medicines that would maybe slow it down for people who are um, uh, suffering from uncontrolled metabolism, right? Or maybe we need to speed it up, right? So that's one example. What else have we learned? Just very recently, right? In lecture 6, we learn a lot of cellular processes in lecture 6 and 7, actually. Um, Anybody know what I'm talking about? Okay, that's fine. Okay, so we talk about like interface is longest one. Yeah, sure. So we understand like cell division, right? So we understand the different phases of mitosis, and we understand when they replicate, we have to copy the genetic material first, right? So by understanding how these processes happens, um, we have a better understanding of how cancer arises, and therefore we can devise plans and drugs that would target to slow down some of these divisions, right? And that would be an example of biotechnology. We understand how DNA replication occurs. If we can control how DNA replication occurs, maybe we can use it to our advantage, and we can, make copies of DNA at will. So that's basically what this lecture is about. Okay, how do we take advantage of some of these cellular processes and use it to our cause. So I'm going to start with what's called recombinant DNA. I'm going to show you a few key biotechnologies in the lecture. The first one is recombinant DNA. Recombinant DNA is basically a term that refers to DNA fragment that has sequences that comes from multiple sources, okay, at least two different sources. So an example is over here, we have the jellyfish at the bottom right here. The jellyfish are very pretty. They have what's called a green fluorescent protein. The name is not important. Okay? The GFP allows them to glow. And so what scientists did, was they extracted the GFP gene from the jellyfish, and then they combine it with the mouse. And now we have these glow-in-the-dark mice. Okay, so these are real, and they are actually quite useful in terms of research. People are not just doing it for like amusement, right? They actually could use these glowing mice for various medical application, medical research. We don't have time to go into all of it. Okay, so that's what recombinant DNA is. Basically, you take DNA from different organisms and you put them together in one sequence, then you have a recombinant DNA. The application of recombinant DNA, I'm going to give you two examples of it. In medicine, it's useful, and in agriculture is also useful. Let's start with the agriculture. Right? There is something called golden rice. Does anybody know anything about golden rice at all? Just wondering. Anybody heard of that before? Okay, well, let me tell you what it is. So in um, countries that are developing, usually these are countries that are poorer. They don't have access to nutritious food, for example. They have what's called vitamin A deficiency. Okay, so with vitamin A deficiency, it leads to preventable childhood blindness, right? Because vitamin A is needed for some of the cells in the eyes to work, and when you don't have enough, you actually don't make a specific molecule, and that molecule is essential for seeing. Okay, at the beginning, it would be just nighttime blindness. In the day, it's fine, but as the condition progresses, you might lose the vision completely. Vitamin A deficiency also lowers immunity. And if you combine that with low vaccination rates, which is usually the case for some of these developing countries because of poor infrastructure, for example, that could spark endemic for infectious disease. In some countries, each year is estimated to kill 670,000 children under the age of 5 and cause an additional 500,000 cases of irreversible childhood blindness. So vitamin A deficiency is quite a serious concern for some of these developing countries. Not so much for North America. You can see green means it's under control. Okay, like it's not even an issue. Yellow, it's quite severe, and then when it's red, it's like clinically uh, 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 severe, important. Right, so you can see it's again in these developing countries. So how do you fix the problem? Well, that's where golden rice comes in. Golden rice looks like this, and it is a genetically engineered rice that is created to make what's called beta carotene, and that is the building block for vitamin A. Right? Beta carotene makes the rice have this color. Where else can we find beta carotene? You might have heard of it before. What kind of vegetables has a lot of beta carotene? Hmm. Carrots, right? You might have heard of people say, "Oh, eat your carrot; it's good for your eyes," right? That's kind of like the logic behind it. Okay, I don't know if it's true, but certainly it has some beta carotene, and so they engineer the beta carotene to show up in the rice. That's not something that rice naturally produces, right? So how do they do that? Well, to make beta carotene, the biosynthesis, right? The biosynthesis means to make the beta carotene. They took a gene called PSY or Psi from daffodil. Daffodil is the flower over here. And they take another gene called CLTL from soil bacteria. And then they put it together, and then they grow the rice from this recombinant DNA. And in the end, you have the golden rice. Okay, so the rice is very useful in combating the vitamin A deficiency because now you just have to eat the rice. It's easy to ship the rice to these places, and in a lot of these countries where vitamin A deficiency is a serious problem, rice is a stable food for them. Yeah. So the proponent of golden rice, people who support the idea of golden rice, is that it addresses the vitamin A deficiency problem, it improves public health, and it also enhances food security. But not everybody agrees with this kind of technology, okay? Because at the end of the day, people say these are GMO, right? Genetically modified organisms. And the opponents of golden rice argue that there might be unintended consequences. Like you don't know long term whether these genetically modified things are going to do any harm either to the person consuming it or to the environment in some cases. Because when you grow it, like if it gets into the wild, like what happens if other animal consume it and so on and so forth, right? There's also concerns about corporate control and patents. Obviously, these are very powerful biotechnology company or sometimes pharmaceutical companies that are involved in creating some of these products. And so, if they patent everything, then there's a cost associated with it, right? Then maybe it becomes like a affordability issue, right? Maybe the countries cannot really afford it, and maybe they're not going to be giving it out for free indefinitely, right? So there's also a socio-economical factor. So that's an example of how recombinant DNA that contains multiple sources. In this case, there is the rice, there is the bacteria, and then there is the daffodil, the flowers. When they all put together, you create something like golden rice, which is useful in both agriculture as well as medicine. Does anybody have any questions so far? Okay, excellent. Now, next we're gonna look at another example, and this is gonna take very long. Okay, this is gonna probably take a majority of the class. And we're gonna look at how recombinant DNA is useful for mass producing insulin. As you might know, insulin is a protein, it's a hormone, so well, it's a protein hormone, okay, that is made in the pancreas. And for people who have type 1 diabetes, these are diabetes that are happening earlier on in life because of the immune system attacking the pancreas. Okay, so it's not the diabetes related to obesity, that's type 2. Okay, so in type 1, they don't produce the insulin, and there's no way for them to regulate their own blood sugar. The only way to help them is to give them insulin shot. Okay, so insulin was first discovered by two Canadian scientists, Frederick Banting and Charles Best. They are shown here in this picture. I'm kind of in the way. Let me move myself out of the way. There you go. There is actually a building dedicated to them, named after them at downtown U of T. If you ever go there, you can check it out, I suppose. So they discovered the insulin, but prior to genetically engineering bacteria to make a lot of insulin, the only available source of insulin was to take it from baby cows as well as pigs. Okay, so they have to take the pancreas out of these animals, they have to ground it up, and they have to extract the insulin, and then they give it to people who needs them. So what are some disadvantages of doing that? Like, what are some uh, drawbacks and challenges of taking insulin from animal source and giving it to humans? What do you guys think? Yeah, so it could be unstable, it could be expensive. Yeah, it's a very laborious process, right, to harness it from these animals. And after you take them out, it might not be stable for very long. Right? So there's a cost associated with it, and there is a shelf life, right, associated with it. Anything else? Maybe just one or two more um, things that you can think of why this might not be the ideal situation. Yeah, allergic reaction, right? People could be allergic to some of these animal products, and if it's taken from pigs, then not everybody can receive it, right? Uh, because of religious belief, right? Some people they do not consume pig, so like a pig product would not be suitable for that reason. Okay, so that's not the best thing, but that's what the only thing that they could do back then. But eventually, in late 20th century, they were able to basically turn bacteria into little insulin factories. Yeah, and then they could pump out, they could produce a lot of insulin. And that's basically how insulins are made these days. So what I'm gonna do for the probably rest of the class is to show you exactly how the process works. Okay. So there are 4 steps involved here. The first step, we have to locate the gene of interest. Okay, there are 46 chromosomes in the human body. Like, where is the insulin gene? Which chromosome is it on? Okay. Once we identify the location, like which part of the chromosome is it on, right? So when we figure out where it is, we're gonna clone the DNA. Okay? We're gonna make as many copy of the insulin gene as possible. Then we're gonna insert it into a vector. A vector is something that would deliver the gene to the bacteria in this case. And then once the bacteria has taken up the vector, then nature is going to do the rest. Okay, so this is an agar play, agar, A-G-A-R, agar plate. It has all the nutrients that are necessary to grow bacteria on them. All these little the dots that you see, those are bacterial colony. Right? There are like hundreds, if not thousands, of bacteria in this little blob. Right? And some are blue, some are white, and I'll tell you why they are like that later on. Okay, so 4 steps. Let's start with the first step, locating the gene of interest. That in the past was basically like virtually impossible. Okay, but nowadays is very easy because of the Human Genome Project, the HGP, Human Genome Project. This was an international collaborative research program with a goal to completely map out all the genes, all the ACTGs in the human body, in the human cell. And it started on October 1st, 1990, and it was completed in April 2003. So basically, it has been completed for a little over 20 years now. And all the information is available online, and it gives us the ability for the first time to basically read nature's complete genetic blueprint of how to build a human being. Well, not for the first time anymore, okay? Like 20 years ago, it was for the first time, but now, we're well into this, and it's quite established. And so there's something called the genome browser, which let me just click online here. Give me a second, it's coming. There we go. Okay, so I already key in a gene here. Okay, the gene I key in was BRCA1. You guys remember BRCA1? It's the breast cancer tumor suppressor gene, right? Yeah. But you can key in any gene that you want. Okay, you can search up like insulin. You can search up telomerase. Okay, you just type it in, and then it will take you to the page. And right away, you can see the BRCA1 gene is going to be chromosome 17, and it's Q21.31. So what that means is this whole thing is chromosome 17. Okay, and right here, that's the centromere. And the shorter arm is the P arm, and the longer arm is the Q arm. So the location is it's right here. It's in a location called the locus, right? The locus of a gene. It's Q21.31. You don't really have to know what that means, okay? But in terms of the base pair, okay, it is going from it's on chromosome 17 base pair 43. Am I reading this right? 43,044,295. Right, to 43,125,483. That's how long the gene is. Okay, like the first letter on the gene, that's like nucleotide 1, right? And the nucleotide 2, 3, all the way. If you go and count all the way, when you get to the 44,295, sorry, 44,295 nucleotide, that's where the BRCA1 gene begins. Okay, so I'm gonna click on it. And there are a bunch of stuff here, but I'm just going to click on the genomic sequence here. There are some things you can change, but I'm not going to change it. I'm just going to click submit. And there you go. This is the gene. This is the gene for the code for BRCA1. And look at what it is starting with. ATG. Right? If you do transcription of ATG, what do you get? This is the coding strand, by the way. If you do transcription of ATG, what do you get? Come on, you can do this. What is ATG? Nobody knows ATG. What does ATG become when you do the transcription, huh? Okay, this is heartbreaking, but it's going to become AUG, right? Okay, AUG is the start codon, which marks the beginning of the gene. Look, there it is. And then we're gonna, you can see this. This is the whole gene. Right? It's just a bunch of A's, C, T's, and G's, right? All the way, pages and pages of it. This whole thing keeps on going, 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 going. That is the BRCA1 gene. Okay, so it's available online. You can search up any gene. If researchers want to look up where a gene is, they could do this with the genome browser. All right, so that's it. Next step, after we located the gene, we have to clone the DNA. We have to make many, many copies of it. And the way we do that is to use the technique called PCR, polymerase chain reaction. And this allows a specific segment of DNA to be copied many, many times outside of a cell. There's no cell involved here, and you can do it very quickly in a very short period of time. Literally in an hour, you can go from one copy to billions of copies of DNA. Okay, so let's take a closer look at how PCR works. To do PCR, you have to buy a machine. Yeah, and the machine allows you to do multiple PCR reaction at the same time, meaning you don't just have to multiply one sample at a time. You can multiply, literally over here. I'm looking at like 30, 40 samples at a time. Yeah, and each cycle of the PCR doubles the number of copies of DNA, and so 30 cycles is going to give you billions of copies. So let's say you start with one copy. One cycle later, you're going to have two copies, and you do it again, you're going to have four copies, 8 copies, dot, 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 dot. So if you do it 30 times, you can plug that into the calculator. You'll see that you will have billions of copies. Okay? So how does PCR works? Like, sure, there's a machine that does it for you, but you still need to, put in the ingredients and stuff like that, right? So how PCR works is based on our understanding of DNA replication. So in lecture 6, we learned that DNA separates by these enzymes called like helicase, and then there are some enzymes called gyrase that would relieve the tension. So instead of using these enzymes in PCR, we separate a DNA strands using heat, okay, around 95 degrees. But the actual temperature might vary depending on your DNA sequence. If your DNA sequence has a lot of A's and T's, then maybe you don't need to heat it up to as high of a temperature, okay, because A's and T's have only 2 hydrogen bonds, right? But if your sequence has a lot of G's and C's, then you might need to crank it up a little bit higher. But let's say around 95 degrees. That's enough to break the hydrogen bond, causing the two strands to separate from each other. Once the strands are separated, each strand can be used as a template to build complementary strand. Okay, so then you use a template to create more, essentially. Now in PCR, the DNA in DNA replication, we have to have an RNA primer, right? The RNA primer is going to basically tell the polymerase where to start copying. So instead of using RNA primer in PCR, we're going to use DNA primer. And you have to design your own primer. Okay, you have to figure out, because we already know where the is located, where the gene is located, you would design primer that would be flanking the gene. Okay, in other words, you're telling the polymerase to copy from here to here. It's like this. Here I have a textbook. Okay, so the textbook is like the whole chromosome. And then I want people to help me copy a few pages. So to tell them where to copy, I put these like, sticky tag here. So, start on this page. Okay, start on this page, and then you copy, 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 copy until you get to the other tag, right? So the tags are kind of like the RNA primers, right? It specify which part of the chromosome you want to copy. Okay, you cannot copy the whole thing; it's too long, right? Like hundreds of millions of base pair, you won't be able to copy all of it. So you just select a short part, and then you copy everything between the primers. Now, in the DNA replication, we have the DNA polymerase 3 that will help us do the copying. However, we cannot use DNA polymerase 3 because we have to heat up the sample to 95 degrees, and at such high temperature, the DNA polymerase 3 is going to denature, is going to break down. It's not even going to be able to do any copying for you. So instead, scientists have discovered something called tag polymerase. These tag polymerase are taken from bacteria that lives in these hot springs. You guys might have seen these pictures before, right? The color is basically due to the mineral deposit, but these are like very hot springs. They go up to hundreds of degrees, right? It's like you can see it bubbling and boiling. And apparently bacteria, some form of bacteria can live in there, okay? And so they extracted the polymerase from these bacteria. They are called tag polymerase, and we can use it in PCR. Okay? So the high temperature doesn't bother them, right? So basically it looks like this. This is the DNA template. This is the section that you want to copy. So you create the primers, okay? And the tag polymerase will continue on from where the primer is. And then it will make the whole thing and make the whole thing. And so after one cycle, you have 2 copies. So here in the PCR reaction, there are basically 3 steps. The first step is denaturation. Again, we heat up to 94 to 96 degrees. These numbers, like, you don't have to memorize them. I mean, when you actually do this thing, you really have to do the math to calculate what the right temperature is. But the idea is you have to heat it up, okay? You heat it up, and this heating process, the denaturation, the heat separates the parent strands. Okay, that's the first step. Separates the parent strands. So you can see over here, step 1, you heat it up. Now they separate. Now all this thing you're gonna do in a tiny tube. Okay, let me show you what the tube looks like. It's really hard for you to visualize, I suppose. But here it is. Okay, it's called Eppendorf tube. Okay, I guess I have to accept the cookies. There are the Eppendorf tubes. Okay, it's really small. It's about this small. And then you would have your DNA sample in there, and you will have all your ingredients in there. Okay, the tag polymerase, the primers, and all that stuff. Okay, so you put it all in, and then you put it in the machine, and you program the machine so that it's going to heat it up. Okay, so once it heats it up, the strand separates. Then you need to cool it down a little bit. Okay, so this cooling stage is called annealing. Okay, the cooling allows the DNA primers to bind. Okay, it's only the primers will bind. The DNA strands are not going to come back together because the primer is relatively short, right? So there are only a few hydrogen bonds there. Certainly much fewer than the template strand. So if you lower the temperature a little bit, it will just be low enough for the primers to attach, but not for the strands to come back together. And then you want to heat it up a little bit again. Okay, not too much, just a few degrees higher. This will activate tag polymerase and begin the copying. Okay, remember these tag polymerase are taken from hot spring bacteria, so they won't turn on unless it's hot enough, right? So you crank it up to 72. Now you might wonder, hey, how come they didn't get turned on at the beginning when you heat it up all the way to 95 degrees? Well, they could be turned on at that point, but at that point there are no primers. Without the primers, they don't know what to copy, right? It's like the book without the sticky tags, right? They don't know which part to copy, right? So it doesn't matter. It's only when you bring it down to about 68, then the primer is going to attach, and then you activate the tag polymerase, then they will copy. And so at the end, you will have like, 2 copies, and each of these copies will go through another cycle of denaturation, annealing, and elongation, and then you're going to have 4 copies. and so on and so forth. Every time you go through this, it doubles. So you just set the machine, you input the temperature for the 3 cycles, for the 3 steps, and then you tell how many cycles you want to run, and then the machine will do everything for you. Okay, that's it. That's PCR. Any questions? Fantastic. Okay, so what are we trying to do? We are trying to create a recombinant DNA so that we can mass produce insulin. Okay, so that we can mass produce insulin. So to create a recombinant DNA, we need 2 sources of genetic information, right? So we got the gene from the human. We make billions of copies of it already. And then the other source would be the bacteria, as you will see in a bit. But let's do a question, okay? I'm gonna launch the poll. This is my favorite part of online learning. It's I don't have to check your 1, 2, 3, 4 fingers anymore. You can just, key in the poll electronically, and hopefully we'll get more participation. By the way, I'm just taking attendance here. Who's Timmy? You're probably using someone else's device, right? Can you tell me who you are so I can mark you present?
My name is Milay.
Oh, okay, that's you. Okay, gotcha.
Yes, thank you.
Thank you. Okay, is that all? I only got like 9 responses. Oh well, we gotta move on. Uh, I was hoping you guys would be more, participate more when it's online like this, anonymous. So a lot of people chose B, C, and E. Let's see. So if you start with 10 copies, right? 10 copies. Right? After 1 cycle, that you times 2, 2 cycles, 3 cycles, and 4 cycles. So what is that? That's 160. That's all. Yeah, that is all. So basically, it's like starting copy, starting number times 2 to the x, 2 to the n, you can say, right? Where n equals to number of cycles. Where n equals to the number of cycles. Okay. Now, PCR, you probably have heard of it during the pandemic, because you can do like a PCR test to see if you have COVID or not. The ones that you do at home, those are called antigen tests. You're testing for proteins in those situations, viral proteins. But the PCR test is testing for viral genetic material. So the kind of PCR that you do for COVID testing, it's called RTQ PCR. RT here stands for reverse transcription, and Q stands for quantitative. Okay, so it's still PCR, but then there are 2 extra elements to it. Right? There's the RT part, and then there's the quantitative part. Now you should know by now. I have been telling you this like the whole course. If you start with DNA, it's going to become RNA, and that is transcription and then translation, right? But viruses comes in two parts, okay? Two types. Okay? There are DNA viruses like chickenpox, and then there are RNA viruses like COVID. Okay? So RNA viruses, when they go into the cell, they actually have to change the RNA into DNA first. And that process is called reverse transcription. Reverse transcription. So when you do your nasal swab, okay, you swab your throat and whatnot, and let's say you have the virus in there, then the swab is going to contain the virus RNA. PCR does not work on RNA. So you have to add an enzyme called reverse transcriptase that would convert the RNA into DNA first. And once you convert it into DNA, you can design primers that will recognize the viral DNA and make many copies of it. Okay, and as you are cloning the viral DNA, there is something that you can add to this mixture that causes it to glow with the DNA. So the more DNA you have in the solution, the brighter it's gonna glow. Okay, so that way you can keep track of how many copies you have in the sample as the PCR process is happening. Okay, so that's why it is called quantitative. Okay, because you can measure how many copies you have as you go, and then you get something like this. Right, this is a curve. So on the y-axis, it's how much it's the glowing is called fluorescence. Okay, so it's how much glowing you are able to detect, and they are graphing it using a log scale. That doesn't really matter. Okay, and then on the x-axis, that's the number of cycles that it's going through. Now, what's important to see from this graph is that each line here is a sample. Okay, so this might be like a patient 1 and then patient 2, okay, patient 3. So each line is a person. Okay, and you're keeping track of how many copies we have in the vial, in those Eppendorf tubes that I showed you, right? As time goes on, and you want to figure out how long is it going to take to reach this particular threshold. Okay, so like I'm making this up. Let's say how long is it going to take for you to reach, like, 10,000 copies. Okay, 10,000 copies. That's what this red line represents. Okay. So you see, for this particular person, for the green line here, this patient, it takes about, 12 cycles to get to 10,000 copies. Okay. For this person, it takes about 25 cycles. 25 cycles to get to 10,000 copies. So I want to ask you, which of these people are more infectious? Is it person A or person B? Who has more virus in them? Is it person A or is it person B? What do you think? Okay, Nadeef said B is more infectious. Why don't we do another poll here? Let's just do a quick poll. Who is more infectious? Is it A or is it B? Who has more viruses in their body? Okay, anyone else? Just waiting. Okay, very good. Well, the more infectious person is actually person A. Okay, person A is more infectious because it didn't take very long. Oops, I don't know why I launched the poll again. It didn't take very long for them to reach 10,000 copy. Only 12 cycles. You only have to multiply it 12 times, and then you only have to go through the PCR rounds 12 times, before it reaches 10,000 copies. Right? Whereas the other sample, it took longer, right? That means it didn't start off with too many copies. So as a result, A is more infectious. So these kind of informations are important early on in the pandemic. Nowadays, people don't really care anymore. Like, you get COVID, you stay home, but they don't really keep track of you anymore. But at the beginning, it was important to keep track of, who's patient 0, right? And who's spreading to who. So that's what qPCR looks like. All right. Do we have any questions before I move on? Anything at all? Okay, nothing. That's fine. Hold on, let me just finish writing down who's here, and then we are done. Okay, good. So next, we're going to talk about the plasmid. Okay, so plasmid are these circular DNA that are found in bacteria. Okay, so bacteria has their own core DNA, which is shown here in red, but they also have these additional circular plasmid that gives them special properties. Okay, so like it might give them like an extra cell wall to protect them. It might give them little hair to stick onto surfaces better so you can wipe them off. It might give them antibiotic resistance, right? And I think I told you before that the bacteria can actually exchange these plasmid. So let's say we have bacteria A that's resistant to antibiotic 1, and then we have bacterium B that's resistant to antibiotic 2, and they could come together and exchange the plasmid, and afterwards you have two bacteria, both are resistant to antibiotic 1 and 2. Okay, so plasmids can be acquired from other bacteria, or sometimes they could just pick it up from the environment. Okay, so let's say like another bacteria died and then it breaks down and its genetic material is spilling out. Another bacterium can come along and then take up the plasmid. Okay, and what we can do is we found out that we can actually take a plasmid and we can insert something in the plasmid. We can insert that insulin gene into the plasmid, and then we can feed it to the bacteria. Then the bacteria will start making whatever's in the plasmid. If we insert the insulin gene in the plasmid, then the bacteria is going to be producing insulin for us. Okay, so that plasmid that we create, the plasmid with the gene in it, that's a recombinant DNA because the plasmid is of bacteria origin, and then the gene is from human origin. Two different sources. Okay. Now, how exactly do we put the gene into the plasmid? So to understand that, we have to talk about something called restriction enzymes. Restriction enzymes originates from bacteria themselves. Okay, and their job is to cut DNA sequences at specific sites. These are called recognition sites. Most recognition sites are not too long. They are 4 to 8 base pair longs, and they are usually characterized by what's called complementary palindromic sequence. Complementary palindromic sequence. What does that even mean? Well, a palindrome in English, it's a word that spells the same forward and backwards. So like madam, right? M-A-D-A-M, if you spell it backwards, is exactly the same thing. So that's a palindrome. Level, wow, kayak, these are all palindromes. You can even have a palindromic sentence. Right? Was it a car or a cat I saw? If you read it backwards, it's the same thing. Okay, so that's a palindrome. Now you can have a palindrome in a DNA sequence as well. If you take a look at the sequence at the top, it's GAATTC. And if you read it backwards on the other side, it's the same thing, GAATTC. So these restriction enzymes, they are found in the bacteria, and they want to cut up foreign genetic material. Let's say a virus, like those bacteriophage, right? Let's say they injected the viral DNA, then the restriction enzyme is going to cut them up. It's not going to cut the bacteria's own genetic material. It's not going to cut the plasmid. because those things have like special markers on them that tells the bacteria, hey, these belongs to us. Don't damage them. Okay. So we can extract these restriction enzymes, and we will use it in the biotechnology. So the restriction enzyme will find these sequence, and then it will cut it up like that. Right. So here are a bunch of restriction enzymes. You don't have to memorize them. If I ask you about restriction enzymes on a quiz or test, I'll give you the information that you need to finish the question. Okay. So anybody know what this bacteria is? Like, this is a very long name, but do you recognize what that is? You must have heard of it before on like, the news or radio or whatever. Anybody know what that? Yeah, that's an E. coli. Very good. So the E, that's what the E stands for. It stands for something that nobody can pronounce, right? But that's E. coli. Okay. When you extract the restriction enzyme from E. coli, the enzyme here is called EcoR1. EcoR1. And it recognized this particular palindromic sequence, GAATTC. And the arrow tells you how is it going to be cut. It's going to cut like this. There we go. Okay. Here is another bacteria that, I haven't heard of it. You might not have heard of it either, but doesn't matter. The enzyme that we get from it is called small one, and it recognized GGGCCC. Okay, and then it cuts it like that. Okay, and then so on and so forth. So each restriction enzyme has its own recognition site, and it's only going to cut the DNA if it has those sequences in it. Okay, if it doesn't see the CCCGGG, then the small one is not going to cut anything. More importantly, you will notice some of them cut zigzag, others cut straight down. If you cut them straight down, like this one and this one, okay, these are called blunt ends. Blunt ends. They are less useful in what we are trying to accomplish here. And these ones that are being cut like zigzag, okay, those are what we call sticky ends. Sticky ends. Sticky ends. So sticky ends are useful for inserting a gene into a plasmid. They are called sticky end because after you cut them, they actually can stick back together. Okay, think of them as like Velcro. You know, Velcro, like those things from the shoes, right? The ones that it's easier than shoelace, right? So when after you cut them, because the AATT is complementary to the TTAA, they actually can come back together and stick back together. That's why we call them sticky ends. Does anybody have any question about what restriction enzymes are for? What are they? And, the palindrome and things like that. Just gonna give you a moment. Okay. Here is an example. Here's an example. We have, could you say again what they are? Oh, sure, what they're for. So the restriction enzymes, they are protection for the bacteria. Okay, so when some random DNA comes into the bacteria, like a virus DNA coming into the bacteria wants to cut it up, right? It doesn't want the genetic material to interfere with its own. So the restriction enzymes would cut up all these viral DNA. That's what it's for in nature. Okay. But after humans discover them, right, we are using it to insert a gene into a plasmid. But I haven't tell you how that happens yet. Okay, I'm going to tell you next. Okay. Is that better? Okay. Now there's a mistake here. I am sorry about. Oh, actually, no, there's no mistake. Okay. I thought there was a mistake, but here it says, there's this is small one, and we're looking for GGGCCC. GGGCCC. Okay, so remember this is 5'. So I'm gonna start looking on the 5'. And then I'm gonna scan the DNA, and we're gonna find GGGCCC. And then we're gonna cut it. Okay, how many times can we cut it? Okay, I get some direct message here. 2 cuts, that's right, 2 cuts. And there is the GGGCCC. You see? So you're gonna cut this off. That's 1 cut. And then you're gonna find another GGGCCC. Okay, tell me to stop. Okay? Or like you can say stop in your head. Okay? Do you find another GGGCCC? Okay? Like right here, right? So you're gonna cut that again. That's it. Okay. So if we get the small one, we can like extract it from the bacteria, right? And then again, everything happens in those little tubes, okay? Those Eppendorf tubes. So you add the DNA sequence, and then you add the enzyme, and then you wait, and it's gonna cut it up twice. Pretty easy, right? You would like that question on the exam, won't you? Don't worry, it will be. Identify the palindromic sequence in the following DNA fragment. Now, this one is a little bit harder than to do. I'll do one for you. Okay, so like I had to read it. Like you don't have to write out the missing strand, but over here we can see it. GCGC. Well, GCGC looks promising because if I write out the complementary strand, excuse me, we see that forward and backwards are the same. So that is a palindrome right there. Okay. Why don't you go find another palindrome for me? Okay, I'm waiting. Give me a palindrome. Oh, I got a message. Was it AGGA? Not quite. Yeah, not quite. Yeah, okay? You figured that out. Okay, that's fine. Anyone else have anything for me? Okay, I'll do another one. Okay. So look over here, right? TTAA. AATTTTAA, right? And then I can even go one more. I see a G here, so I could go C. I see a C here, I can go G. Right? Palindromes are always even number of letters. Surya said TTTAAA. Absolutely. Right? And I can actually do another one too. Right? So that's it. Those are all the. Oh yeah, you found another one. Oh, well done. Okay, that's it. Not AAGG though, right? Because AAGG backwards is CCTT, right? Trevina? Okay, that's good. Let's try another one. Go ahead. Okay, I'm looking for how many fragments are we gonna produce? I'm looking for a number here. Hey, I hear a 3. Anyone else? Okay, don't give me the sequence. We're not talking about the sequence here. We're talking about how many fragments are going to be produced. Right? So how many times are we cutting this, in other words, right? And then you will have to see how many fragments. So let's say we look at equal R1. Oops, equal R1 is right here. And equal R1 is going to be cutting GAATTC. GAATTC is right here, right? So that's going to be one cut. GAATTC. Is there another GAATTC somewhere? Another GAATTC? Right, like right here, right? So that's another cut. And so that's 2 cuts. And then what about Hindi3? That's how you pronounce this one. Hindi3. It's AAGCTT. There it is, AAGCTT. So that's gonna create another cut. Oops. So how many fragments do we have? Well, think of a line, right? If you have a line and then you cut it 3 times, you're gonna get 4 fragments. Fragment 1, 2, 3, and 4. Yes, this is gonna be recorded. So 1, 2, 3, 4. The answer is 4, okay? 3 cuts, 4 fragments. 3 cuts, 4 fragments. Okay. Next, we're gonna take a note over here. Okay, so this note, we're gonna explain how to inserting a gene into a plasmid. Inserting a gene into a plasmid. All right. So here's a plasmid. And you can buy plasmid online nowadays. I mean, you can buy anything online nowadays, but then like there are like biotech companies that sells these plasmid, okay? You can buy them. And to keep it simple, I'm gonna say that there is a restriction site here. AAATTT. So on the other side is TTTAAA. Let's just make this a little bit bigger. There we go. And let's just say this is the recognition site of restriction enzyme 1. Okay, we'll call it like RE1, restriction enzyme 1. Okay, so just gonna give you a moment to catch up with the drawings. So let's say you buy this thing again online, and it comes in a little vial like this. Okay, those tube things that I showed you, the Eppendorf tubes. So you know you're gonna have the plasmid in there, thousands of them. And so what we're gonna do is we're gonna add the restriction enzyme 1 to it. And you're gonna add the restriction enzyme 1 to it. And so what the restriction enzyme will do is that it will cut it up. It will cut it up. Right? And I'm just making this up. No, I think I have an extra one here. Yep, there we go. There's an extra one here as well. Sorry. So we'll cut it up like this. And I know it's difficult to see like that, so I am gonna put a little outline around it, okay? So you cut a zigzag, and basically you pop open the plasmid. And what do we call these things? What do we call them? Do you remember? Does anybody remember? They are fragments. Yep, they were recognition site, but then I didn't cut them straight. I cut them zigzag, so they create those things. What do you call those things? That can, come back together. Sticky ends. That's right. Okay. And you can see why, right? I mean, I cannot show you on, on the computer, but you can see how you can like glue them back together, right? Because the A here wants to connect with the T, and then the A wants to connect with the T, and then the TT want to connect with the AA, right? Do you understand that? So the ring might actually close back together. So that's that. We're gonna leave it at that for now. Okay, so we have a tube here with a plasmid with the restriction enzyme 1, okay? And then now we're gonna have another tube. We're gonna have another tube here. This is from the PCR. This contains the gene of interest. Cloned using PCR. You've copied it many, many times already. And so let me just draw the gene for you. This is one of those copies. It's like ACTGs, right? In it, whatever the base pairs are. And then at the end, you actually have what we call like a flanking sequence. Flank is like on the side, right? So, in this hypothetical situation, the flanking sequence might also contains the same recognition site. Same thing over here. Okay, so these are flanking sequence. Contain the same recognition site for restriction enzyme 1. Okay. Now, of course, that's not always the case. And when you actually do this stuff, you have to check what the flanking sequence are. Maybe it has a different restriction enzyme site. And maybe it's for restriction enzyme 5, right? Then you have to find a plasmid that also have restriction enzyme 5 recognition site in it. The bottom line is the recognition site flanking the sequence has to be the same recognition site as the one that is found in the plasmid. Okay, so you're gonna take this guy, and you're gonna add restriction enzyme 1 to it. And then what's gonna happen is you're gonna cut it. Okay. You're gonna cut it, and you will end up having something like this. And then cut it on the other side as well. So you have sticky end here. And then you have sticky end. Okay. The other half just kind of falls off. Okay, like, you know, we don't really need them anymore. Okay, so this is the gene of interest. Of course, you have to make sure also that the AAATTT does not show up inside the gene. If it does, you're gonna end up cutting the gene as well, right? So we don't want that. So now we have the same kind of sticky ends in the flank as the plasmid, right? Which means I can actually take this guy. Yeah, let me see if this works. Sometimes it works, sometimes it does not work. Which means I can actually take this guy. Okay. And we're gonna put it in. Nope, totally does not work. But you can kind of see it, right? You can see how the sticky ends are complementary, right? So basically, we can take this gene and add it with the plasmid, and the gene is gonna go into the plasmid. Do you understand? Like how this end is gonna go with here. You know what? Let's use a highlighter. Like this end in the plasmid is gonna stick to this end of the gene. And then like this end over here is gonna stick to this end. So essentially the gene will go into the plasmid. Any questions for that? Okay, so that's what's important. That's why you have to treat them with the same restriction enzyme. Right? If you treat the plasmid with restriction enzyme 1, but then you treat the gene with restriction enzyme 2, maybe you create sticky ends in both cases, but they wouldn't be compatible. Like if this was like a GGCCC, it's not going to be able to stick to the TTTAA, right? Okay. So finally, you mix them together. Mix the two together. And then you add glue. Okay. So then you have the gene that goes in. And this is the plasmid. What's the glue that sticks things together in DNA? Anybody remember the name of the enzyme? That would stick it together, huh? What is it? No, I'm looking for an enzyme. Good try though. You know, at the end, when you need to glue the fragments together, what enzyme do we add? Ligase. Okay. And then you have it. That's it. Then you can feed this to the bacteria. And then the bacteria is going to make whatever the gene codes for. If it codes for insulin, it will mass produce insulin. Isn't that cool? Okay. Let's have a break. Okay. Sometimes it's more tiring sitting virtually to hear me than in person. So let's take a 5-minute break. Okay, don't be gone for too long. We will resume at 1:45. Okay, 1:45. I think we'll go on a little longer today. If we finish enough slides today, then I can let you go after the quiz on Tuesday. Okay. We'll see. We'll see how much we can do. Okay. 5-minute break. See you in a bit. Okay, let's keep going. So now that we have inserted the gene into the plasmid, we're gonna have to feed it to the bacteria. But before we do that, we gotta understand a little bit more about what a plasmid is. Okay. So this is a very simplified plasmid, okay? But the ones you can buy like online from like biotech companies, they are usually much more complicated. Let me just show you a map that is I can find online over here. This is a plasmid map, okay? Like it has a lot of information on it. Okay, but let me show you the simplified one first, and then I'm gonna show you that complicated one again, and then you will be able to make some sense out of it. Okay, so over here in our plasmid, we have what's called like the origin of replication. Origin of replication. You know what that is, right? That is where the process of transcription is gonna start. Okay, so that essentially is where the kind of like the promoter is, if you would. Then we're gonna have these things. Okay? PST1 equal R1, BAMH, alpha 1, and these are restriction enzyme sites. Okay? So those are restriction enzyme sites. Okay. So what that means is if you want to use equal R1 to insert the gene, you will insert the gene here. If you use PST1, then you will insert the gene here, right? So it just gives you more option, right? Because I was telling you, the flanking sequence might not have the restriction site that you're looking for, right? In our example, maybe the flanking sequence doesn't have the AAATTT, then you have to find something else that works. Okay, so maybe you have to try like BAMH or you try alpha 1. So by having more than one of these restriction enzyme sites, it gives you more versatility in terms of what you can insert into the plasmid. So this is the plasmid map, and you can see that's the origin of replication. Okay, and all these black things, that's restriction enzyme sites. Okay, so it's very versatile where you can insert. And then in addition, we also have these selection markers. Okay. So TETR stands for tetracycline resistant gene or resistance. This is ampicillin resistant. Okay. So that will give the bacteria resistance to these antibiotics. That's what they are. The tetracycline and the ampicillin, they're antibiotics. Okay. So normally the antibiotics will kill the bacteria, but then if they take up the plasma, when you feed them the plasmid, if they accept the plasmid, they will gain resistance for these antibiotics. So we can use them as selection markers. Okay, it will be useful for us to tell whether or not the bacteria have accepted the plasmid. Okay, I'll explain more on that later. Just write down these things first, and that's just the name of the plasmid. Okay. Okay, so let's take a look here. More notes. All right, so now that we have this plasmid with the gene of interest, we're gonna feed it to the bacteria. So again, the bacteria is gonna come in a tube like that, and you basically feed a bunch of these, you know, to the bacteria. So this is bacteria, and then you're gonna mix the plasmid into the tube. And there are different ways you can encourage them to eat it. Okay, you can shock them with some electricity. You can, shock them with some extreme temperatures, and then that would encourage them to take it. But the truth is, even if you encourage them to take it, they might not all take it. Okay, there is going to be some bacteria that just will not accept the plasmid. Okay, so how do you know? How do you know which bacteria has accepted the plasma? I mean, they look the same. Okay, to the naked eye, you wouldn't be able to tell, right? And that's where the selection marker comes in. So let's take a look here. Okay, let's say we have the bacteria. Okay, some will accept the plasmid, others will not. Okay? And let's say in this plasmid that we were using, in addition to the gene that we have inserted, there is also an ampicillin resistant gene in it. Okay, so let's take a look at what happens. Let's say we take these bacteria after we feed them the plasmid and we grow them on a petri dish, like the one I showed you before. So this is an agar play plus nutrient. Okay, and then we're gonna put the bacteria on the plate and just let them be. It has all the nutrients necessary for them to grow. Okay? So if you do that, you add the bacteria on, do you think they will grow or not grow? Do you think they will grow? Well, it's not a trick question because it has all the nutrients, right? So they're just going to grow. In fact, they will grow a lot. Okay? They will very happy. Okay? They will be very happy. It will grow all over the place. Okay? So lots of growth. Sometimes we call this lawn growth. Grows like the lawn, covers the whole lawn. So this is just a control. This is just to see, if we accidentally kill the bacteria while we try to feed them the plasmid. Maybe you shock them too much with the electricity and you kill them off or something, right? Like you just want to make sure that they are still alive, okay? But the problem with this is, like, by the way, these are colonies, right? It's not a single bacterium. It's like a whole group of them, right? But how do I know which one had the plasmid? How do I know which one accepted the plasmid? Because I only want those. Those are the ones that can make insulin for me. How do I know? I don't. Right? So that's why I need to have another play. There's another play right here. Okay. Another agar play. My goodness. Oh man, one more time. There's another play. And it's agar plate plus nutrient. Nutrients, I guess, plural. And also ampicillin. Remember, ampicillin is an antibiotic. It's supposed to kill bacteria. So if I take the same tube and then I put it on this play, what do you expect to happen? Lots of growth, some growth, no growth, or die. What happened? What will happen? Little growth. That's right. Okay, you will have some growth, but not a lot. Okay, why? Because only bacteria that has accepted the plasmid will survive. Why? Because it has the ampicillin in it. The ampicillin will kill the bacteria unless they have the plasmid, because the plasmid has the ampicillin resistant gene. Right? This gene allows the bacteria to grow in the presence of ampicillin. Okay, so that's how we know all the ones that are growing on this second play are the ones that have the plasmids. Those are the ones that we want to mass produce, because those are the ones that are able to make the insulin for us, right? And again, how do we know they are the ones with the plasmid? Well, they survive. The only way for them to survive is because they have the anti-resist ampicillin resistant gene that comes with the plasmid, right? Okay, please ask me questions. I know some of you must have questions. If you have a question, someone else must have the same question, okay? You can private message me if you don't want the whole class to hear, or you can just ask. Okay. Let me know before we move on. Really? Everybody understood that? Okay, well, that is that. Can I run it back again? Like the whole thing, or like which part? It's better if you ask me a specific question, right? Because if you want me to explain again, then you can just watch the recordings, right? But if there is a specific point that you don't understand, then I can. What is the word after the grow in the in the presence? Yeah, sorry. Grow in the presence. You mean this one, God power? Is this the one that you like? Is it that you don't understand why they grow, or what is it about this that you don't understand? You don't understand why they grow, or you don't understand why they are the ones that we want? Okay, so basically, again, right, like this play is different from the control play. Why they are the ones we want? Okay, yeah, we want them because these are the ones. The fact that they are able to grow in the presence of ampicillin, the fact that they are able to survive even though you give them ampicillin, which is supposed to kill them, right? The fact that they are able to live tells you they have the plasmid in them. The plasmid that we feed them, not only does it contain the gene we wanted to mass produce, but it also contain the ampicillin resistant gene. Okay, the ampicillin resistant gene allows you to survive even though there are antibiotics. Okay, so if the bacteria did not take up the plasmid, they will die. They won't grow. The fact that they are growing means they have the plasmid. Is that a little bit better? Okay, so we'll do a few questions here. A gene is inserted into the plasmid using E core R1. And so look at the plasmid map. If we insert the gene using E core R1, it is going to go right here, right? That's where the gene is going to be. It's going to pop right in there. Gene. And then you transform into a tube. Transform means you feed it to them. Transform means you feed them. That's just like the proper word. Okay? Feed the bacteria. And then you culture all of them on a plate containing tetracycline, another antibiotic. What result do you expect? Please participate if you can, all right? The poll is anonymous, and the more people participate, the better I will know whether or not you guys get this stuff. Anyone else? Someone choose F. I don't know why. Do you see F on the screen? Okay, I'm gonna end the poll in a few seconds. Okay. Most of you got it. Only a small portion will grow. Only a small portion will grow. Why? Because not all the bacteria will take up the plasmid, but if they did take up the plasmid will give them tetracycline resistance. So even though there are tetracycline in the play, it will only kill off those that don't have the plasmid. Okay? And the ones that have the plasmid, they will grow. Right? So a small portion will grow. Now let's take a look at a slight variation in the question. It's the same thing, but this time, instead of inserting using equal R1, we are inserting using SMA1, small one. And then, small one is right here. And then you're gonna feed this to the bacteria, and then you're gonna grow them. Are they gonna live or not live? Okay, let's try to do this one a little bit quicker. You're gonna grow, not grow, some grow. Okay, last call. A couple more seconds. Okay, so if you insert using small one, the gene is actually going to go right here. It's going to go right here. And when you insert the gene, you actually will destroy the tetracycline resistant gene, right? Because your gene is going to go right in the middle of it, right? So in this case, even if they accepted the plasmid, they will still die when there are tetracycline. Because the tetracycline resistant gene in the plasmid is disrupted by the insertion. So all of it would die. Okay? It doesn't even matter if you have the plasmid or not. Okay? If this is the case, you messed up your experiment. Okay, that's that. All right, let's go back to the study guide here. And I don't know if you have it, but there are a few questions here that we want to take a look. Okay, here is the plasmid. Here's the plasmid. Let me just take a picture of the plasmid here. Here is the plasmid. And I'm just gonna skip to E, okay? Just gonna skip to E. I'll post, I think the video for this, taking this up is already on eCentennial, so you can watch it later, but I just want to go through one more example with you. So you're gonna insert the gene, right? And this time you're gonna insert it with BAMH for this plasmid, okay? So BAMH is over here, right? That's BAMH. You're gonna put the gene right there. You can't really see that. Let's do it again. You're gonna put the gene right there. Okay? So if you grow them with no antibiotics, what do you expect? No antibiotics. Lots of growth, no growth, some growth. There are only three choices. Do they grow or not grow? And if they do grow, is it a lot or a little bit? Yeah, no antibiotic is always the control, lots of growth. Okay, they're happy. They have nutrients. Okay, what if you add tetracycline? If you add tetracycline, are they gonna grow or not grow? Grow or not grow? Not grow. That's right. Why will they not grow? Because the gene is breaking the tetracycline resistance, right? The gene that you inserted disrupted the tetracycline resistant gene. So it doesn't matter if they have the plasmid, it will not protect them. Okay. What about with ampicillin? Do you think they will grow, not grow, some growth? Some, that's right. Very good. Because the ampicillin gene is not disrupted, right? You understand that? Right. So it's not too difficult, I hope. Right. Just go through it again if you need to. And then you know, you should be okay. All right, let's see. Mm, let's see how many more slides we have. Are you guys good to go for a little longer? What's the, let's see what's the sentiment in the class? Okay. Do you think you guys can go for like another 25 minutes or not? Either way is okay. Like A, okay, 25 more, and then no class on Monday after quiz, or B, no more. Okay, what do you want? Majority wins, okay? No, no more means we have to have class after Tuesday, okay? Tuesday after quiz. Okay, anyone else? You gotta voice out. I don't know what you want, okay? Should we go more or no more? Okay, I'm 50-50 right now. I need another vote to see what's going on. Oh, I guess the rest of you don't really care, right? Okay. Well, you know what? I think if half of you are tired, then I think maybe it's a good place to stop. Okay. I don't think, do we have a lot of what does good to go mean? Good to go, like good to stop, or like go more? Go more. Okay, let me see. Go more. Okay, hold on. We are slide 36 right now. 58. Yeah, I think we should stop. Okay, I think we should stop. Okay. And then Tuesday after the quiz, we'll stay a little longer. I promise I won't be too long. Okay, and then we'll wrap this up next week. Yeah, that's it. Okay, thank you for coming online. And please watch the quiz for review. Okay, you must watch it. Watch it a couple of times. Right, and then you'll be ready for the quiz. If you have any questions, feel free to find me. Otherwise, enjoy the rest of your week. Take care, guys. Bye bye.